This video is going to give you an overview of how to analyze your data from the Enzyme Independent Lab activity that we did last week in order to prepare the data for your lab report, specifically the things you'll need for your result section. So before you get started, what you'll want to do is type your data into Excel. For each of your two experimental groups, you should have the label in the first row. So for my pretend experiment, I have chicken liver as one experimental group and ground chicken as the other experimental group. Then you want to have a label to the left of sample 1, sample 2, sample 3, since you should have done 3 test tubes for each of your experimental groups. For chicken liver, each of your 3 samples, you should provide the value either in centimeters or millimeters, however you measured it, provide that value or type that value into the box. It is very important that you do not include units. You only want numbers. You don't want cm or mm. You don't want any letters in that box. You just want it to be the value. So type your values for the chicken liver, type your values for the ground chicken. With these values, the first thing we want to do is determine the statistical information we need. In particular, we want to know the mean or the average of each group, the standard deviation of each group, the count of each group, and then we want to measure the p-value to detect if a difference we see between chicken liver versus ground liver is actually due to that independent variable or if it seems to be random. So in order to answer that question, you need to come up and click the Data tab. If you took Bio 111, you should have already gone through the process to get the data analysis tool on your Excel data tab. If not, you're going to have to work through the steps to add that. This is essential to have. So you're going to click on this data analysis button. It's going to bring up a lot of different options. The first option you're going to click is descriptive statistics. This option will tell us the mean, the standard deviation, and the count. So I'm going to click descriptive statistics and hit OK. And now it's going to ask me to input some information. First, it asks me the input range. So what data does it need to analyze? In order to provide that, I'm going to come over and click this arrow right here. And that allows me to select the information it needs to analyze. So I'm going to click and drag to highlight those cells right there. So my title for each group and then the 3 values in each group. Once again, make sure you don't have units here. I'm going to click this little arrow and it'll bring me back to my box. And now it's told my descriptive statistics what data it needs to analyze. You'll also need to click this button that says labels in first row so that it knows chicken liver and ground liver is the title, not an actual data point. And then you want to come down and click this button that says summary statistics so that it gives us the correct information. Once you do that, you'll hit OK and you should have a new window pop up with a lot of different values. You'll notice the 2 left columns are the values for chicken liver. The 2 right columns are the values for ground chicken. Some of the most important pieces of information we have here are the mean, which I'll go ahead and bold that just for our purposes. So the chicken liver mean is 7. The ground chicken mean is 2.6. I pretended that I measured my data in centimeters. The standard deviation, that tells me how much of a spread do I have much between my values. The standard deviation for chicken liver is 0.26. Standard deviation for ground chicken is 0.3. I also want to know my count, which is pretty simple. How many data points do I have in each set? I have 3 samples for chicken liver, 3 samples for ground chicken. So those numbers will be more important to me in a second. I'm now ready to do my second type of statistical analysis. So I want to come down and click back on sheet 1, which was my initial sheet. I'm going to come back up and click on the Data tab and click on the Data Analysis button. This time, as I scroll through this list, I want to click on the option that says t-test to sample assuming equal variances and hit OK. Once again, it's going to give me a box in which I need to input the information for it to analyze. This time you have to input each experimental group separately. So variable range 1, I'm going to click on this up arrow. And I'm going to select my chicken liver column, just my label and my values. Click that arrow again, and now it's in there. Same thing for variable range 2. I'm going to click that arrow. Now I'm going to select my ground chicken column. Hit that arrow, and now it knows the data to analyze. For hypothesized mean difference, you just want to put a 0. You want to click the button that says labels so that it knows chicken liver and ground chicken are titles, not values. And then you should be able to hit OK. And once again, a new sheet will pop up. Now it's going to give you some of the same values that you had in the previous statistical analysis, but the one that we're most interested in is the p-value for a two-tailed t-test. If you remember from Bio 111, we can measure the significance of data with the p-value. If that p-value is anything less than 0.05, that indicates that the difference between the two experimental groups is significant and truly due to the independent variable, not due to just randomness. Whereas if that p-value is higher than 0.05, it means that our independent variable might be causing a difference, but it may not be causing the difference. So this p-value, you'll notice, is written in scientific notation. So 4.47 times 10 to the negative 0.5. So technically that p-value is 0.0000447. So this is our p-value, and it is definitely less than 0.05, meaning our results are significant. So what I'm going to do now is go back to my original sheet, and I want to create a new table kind of similar to my other one. So in one box, I'm going to type chicken liver as a title. In my next box, I'm going to click or type ground chicken as my title. And then down the side, I want to type mean, standard deviation, count, and p-value. And then I want to type in the appropriate values. So mean is going to be 7 and 2.6. And once again, leave those units off. Standard deviation is 0.26 and 0.3. The count is 3 for each of them. And then the p-value is 0.0000447. Now, one very important thing to note is that there is only one p-value for the entire experiment. There are not two different p-values. One thing that I like to do to represent that is instead of having it sit in one column versus the other, I actually like to highlight both of these cells and then hit this button that says Merge and Center. And it'll actually put it across both cells, which helps to represent that the p-value represents all of my data, not just chicken liver or ground chicken. And so this table that you just created will need to be copied and pasted into your lab report. It should be the table that you have in your lab report in the results section. In addition to the table, you also need a figure depicting the difference between the chicken liver and the ground chicken. In order to make that bar graph, you want to have a bar for chicken liver that indicates the average and a bar for ground chicken that indicates the average. In order to do that, I am going to drag right here, highlighting my chicken liver and ground chicken. Actually, let me start over. I'm actually just going to highlight these 4 cells right here. So chicken liver, a mean of 7, ground chicken, a mean of 2.6. With those highlighted, I'm going to come up and hit Insert, come over to my different chart options. Now I'm going to click this one in the top right corner that, there we go, that looks like a bar graph. And I'm going to hit my first option. So that automatically creates a bar graph for me that demonstrates that my average for chicken liver is much higher than my average for ground chicken. But as you hopefully remember from Bio 111, we need to make some changes to this chart in order for it to be formatted properly according to our lab report guidelines. The first thing we need to do is get rid of the chart title. So I'm going to click on that box and hit delete, and that will go away. I also want to make both of my bars black instead of blue. So I'm going to click on them. You can come up to format, and then this shape fill button, you can click black so that they're both black. I also need to add an axis title to my y-axis so that I know what these values represent. So if I come up to the chart design tab, I can click add chart element, axis title, primary vertical, and that creates a box where I can type my axis title. So if I click on that, I can make this foam production, then in parentheses in centimeters. It might be centimeters for you, it might be millimeters, either one is okay. The last thing I need to do is delete my grid lines. So I want to tap on those grid lines, once again hit delete, and it'll make those go away. So now I have a figure that belongs in my lab report and a table that belongs in my lab report.